So persister cells are really important and difficult cells to treat. And so these are cells that basically escape the therapy. And the problem is to detect these cells, there are only a few of them in the whole tumor. My name is Alexander van Oudenaarden. I'm a physicist by training. And over the years, I became more and more interested in biology. And I recently got interested in PERSIST-SEQ, mostly because of the science behind it, so to understand persister cells in cancer. And my role there is an academic lead. So this is the Hubrecht Institute for Developmental Biology and Stem Cell Research. It's more than 100 years old. It started in the center of Utrecht and moved in the 60s to what they call the Uithof at that time, which is the Utrecht Science Park. And was actually the first building on the Uithof. Since the 60s, it grew out into a huge institute with 2 additional buildings actually. It's focused on stem cell research and developmental biology. Our lab is mostly interested in developing technology for sequencing individual cells. And that's really very useful in developmental biology because during the growth of an embryo, almost every cell is different. So if you would take that whole embryo and you would analyze it on average, you would get the average cell, but the average cell actually doesn't exist. Every cell is different. Recently, we got really interested in cancer as well because there is a very similar problem, particularly for persister cells, is that you have this huge population of cells where there's only a very small fraction of cells that are causing the trouble. And you want to analyze these cells. Putting that into a blender will not help you. You have to analyze every cell separately. One of the major goals of PERSIST-SEQ is to detect persister cells and to understand how these cells are different from the other cells. That's really what my lab is really obsessed with. Single-cell sequencing, which is a method that we're very much excited about, is a very proper technology for this type of cancer research. And that's why we really got interested in this particular project. What we hope to do is to develop new technology that will allow you to really detect the persister cells from all the other cells. And for this, you need technology that can really analyze not the whole tumor as one piece, but each individual cell of the tumor. And that's why we're collaborating with a lot of the cancer biologists to make this technology available to them. We are really working for the last 25 years on technologies that can analyze single cells and try to understand how one cell is different from its neighbor. So that's why I really got interested in this project, because I felt that the technology that we are interested in could help cancer biology. A few years ago, we had a facility as well that turned into single cell discoveries. A master student in our lab and a PhD student, they started a company with 2 people, and I think now it's more than 50. So it was highly successful. So we're very proud of actually having single cell discovery being a spin-off of the Hubrecht Institute. So the PERSIST-SEQ consortium is a very wide spectrum of different scientists. So there are the academic people, but there's also the small companies, and there's the huge companies, and we all have to collaborate together. This is also something that actually I find very interesting because I'm a real kind of basic scientist, and before I got in the consortium, I had no idea how companies were thinking. Everybody has different ways to collaborate, and I'm mostly used to academic collaborations, and I think it's also good to solve a problem like a PERSIST-SEQ problem because the different companies and academic partners think very differently about this problem. So if you think together and combine the best of all the worlds, you actually have a better chance solving the problem in the end. Single-cell sequencing is a fantastic method to really analyze an individual cell. As soon as you take that cell and you analyze it, you know what this cell was doing at the moment that you took it and opened it up. So this is kind of a snapshot of what the cell was doing at that moment. And if you do that for many single cells, you know at that point what this population of cells was doing. This allows you to really get a very detailed view on how cells are communicating with each other. This is very important to do at the single cell level because if you would just put all these cells in a blender and there's like 1% of these cells are persister cells, you won't see them because they will be overwhelmed by all the normal cells that are in there and you would not even know there are persister cells in there. This technology is so powerful for thinking about this problem. We aim indeed to analyze 5 million cells. One thing which is really important is that all these cells were prepared and processed in a very similar way so that all these different locations, all the different scientists that are working on different organs as well, breast cancer or colon cancer, that all these protocols and ways to analyze and sequence the data are uniform across all the sites. So that ultimately you have a huge data set where you can compare all these things without having to worry too much about systematic changes between different labs. We have put a lot of effort in this in the first few years to make sure that we have a protocol and a pipeline of generating single-cell sequencing data that is the same for everybody in the consortium. So what normally happens is that one lab would have their own system, and there's very big differences between how people do this. So it's very difficult to compare those data sets. We therefore want all the sites to do exactly the same thing. And that has been a big challenge to set that up, because people, of course, want to do their own thing. So what we decided is to have one central location where all the data is stored. Single Cell Discovery is a small company that is doing all the sequencing for everybody. So this makes it very central and allows us to really compare all these different experiments in a systematic way. They will sequence the cells, and they're all deposited into a central place where then all the different partners can log into and analyze the data. All the data analysis tools are also there provided. That allows, again, to get a really uniform way to analyze the data. And the same holds for the generation of the experimental data. That's done within Single Cell Discoveries. They do everything the same. It's all at one place. So the only thing that we still have to worry about is how do you get the cells from A to B, and that is also uniform, and that where we have long protocols to tell people how much dry ice do you put in there, how long do you freeze them. And as long as people follow that protocol, everything is comparable. I'm also part of several of the work packages, including work package 2, which is the portfolio management. And it's something that actually I never saw before in any proposal or collaboration. This is a work package that determines which experiments are going to be funded. So what we decided in this consortium is actually to put a large part of the budget in work package 2 reserved. And then the work package 2 will determine, based on small proposals that are written by the different scientists, to actually fund small projects. So you have to apply for that money, and then you can, if it's approved by a committee, you will get this particular funding to sequence a certain amount of cells. So it's I think a very nice quality control built into this whole system. Work package 6 focuses on spatial transcriptomics. We're exploring this a little bit in the consortium. It's really a little bit too early for the consortium to go full force in that direction. We're exploring a little bit, but just at a very low level. The consortium consists of a wide range of expertise, ranging from very technical things like what we're doing, like developing technology, to surgeons who would take biopsies and have material of a real tumor, and anything in between, of course. That's really, really powerful, because this allows you to also compare all the different conclusions that you get from the very technical to the clinical across model systems. Of course, we will also learn detailed things about how a certain form of breast cancer, how these persister cells will behave there. Perhaps because I'm raised as a physicist, I hope that there will be some general principle, something that's valid for all cancers and all systems that we could extract from this. I'm not sure if that will work, because we're really in the process now of collecting all the data and comparing it to each other. After the consortium stops, all the data will be available to the rest of the world, and also all the things that we learned, the conclusions about the data, the interpretation, all the protocols that are going to be accessible to other consortium that will be starting in the future. If we can show that it really works, it might be a blueprint for future consortia that they can use all this knowledge that we accumulate in terms of the technical structure to how to set it up. They just copy that. I think this is something that most people don't think about, but as soon as you have like 20 groups working together, you need to make sure that you can compare all these experiments across the 20 groups, and you need some rules and protocols that are obeyed by all of them, because that allows you then to really compare them one-to-one. So that in industry, this is much more common to do very standardized things. If you combine this with the academic power of generating a lot of new hypotheses, I think it is something very useful for future consortia to follow a similar strategy. So we're thinking currently how to do that best. I think what is really nice is that we already have this very central way of storing data and analyzing data. So it'll be fairly easy to take that whole part and make it available to the whole world.